On? Okay, if not, I'm going to tell you a bit about Bryn while he's trying to connect. Bryn Dentinger is the curator of mycology at the Natural History Museum in Utah. He was head of mycology at Kew Gardens in England for many years, a very young head of mycology. So we're really, really happy to have him with us because he really is, one of the people I admire the most in mycology. So we're going to be talking about the genus Psilocybe. We're going to be talking about how long Psilocybe has been around on Earth. We're going to be talking a bit about what the differences between the genus Psilocybe, the genus Stropharia, and other genera are. And we're also going to take a deep dive into whether psilocybin is only present in the genus Psilocybe, which many of us mycologists know is not the case. So let me try once more to get Bryn on. Okay, and if we can't get Bryn on, we're going to go the other way around, and we're just going to do this on Zoom and put it forward. I beg you to we're just going to go through Zoom, I think, and put this on. So, give me 1 second. I'm connecting with Bryn, and please, I'm just going to log off for 1 second. Bryn, I'm getting through to you on Zoom, and we're going to put this out through this same account in about 2 minutes. Yes, it's because you're trying to connect on your laptop. You have to do it on your phone, Bryn. That's what it is. It has to be on your phone. That's why it's not working. Okay, we're going to get through on the phone. We're not ending this. Here we are. We have Bryn. Hello. Are you there? I want to acknowledge that there are people connecting from Australia, the US, everywhere. Hi, Bryn. Here you are.
Hi.
All the way around. Turn the camera.
So I'm obviously a noob when it comes to social media.
Good evening.
Hi.
How are you? Welcome.
I'm all right. I'm doing okay. Yeah. Salud.
Salud. Welcome to a Myco Viral Night with Julie. I want to welcome you once again. You were here talking about fungus farming ants. I want to let you know that we have people connecting from all over the world, and we have the one and only El Grande, my idol, Paul Stamets, connected too, who shout out to Paul always. We have people connected from Australia, Chile, Mexico, Argentina, Colombia, US. We even have some of your students connected. So,
all right.
Welcome to the genus Psilocybe in the molecular era. I want to start by asking you, what happened? What happened when molecular tools were available? What changed for Psilocybe?
Well, the biggest change that was discovered from molecular phylogenetics was that the genus Psilocybe was not monophyletic, meaning there wasn't a single common ancestor that they all shared, but instead there was more than one, and so the genus got split in a small group that were once classified in the genus Psilocybe, which are the non-hallucinogenic, non-active group. They got carved off into its own genus called Diconica.
So inside every species in the genus Psilocybe has psychoactive properties?
As far as we know, that is true. Yeah, in the currently circumscribed genus Psilocybe, those are all probably active. Now, not everyone has ever been surveyed for these chemicals, at least not by scientists. So we don't know for sure. But from what we know about the distribution of the chemicals in the species that we know about, they probably all contain them.
And what happens? You mentioned that the non-psychoactive species that were once called Psilocybe were carved off to a new genus called Diconica. But there's also the genus Stropharia that was, toyed around. So Stropharia cubensis, Psilocybe cubensis. What happened to Stropharia?
Stropharia still exists. It's just that it's a much more restricted definition than it used to be. So we, I don't know how familiar your viewers are with fungal taxonomy, but it's gone through a revolution in the last decade, and we've discovered many new things about relationships that have resulted in a lot of changes, like the Stropharia example, and that's kind of shaken things up across the kingdom.
So before we would name a fungus based on its microscopic features paired with its macroscopic features, and today it's relationships that define who one is. Is that correct?
Well, I think it's always been, since Darwin, it's always been, the assumption has been that our naming system reflects ancestry. And up until 20 years ago, we didn't really have access to information about these organisms other than how they looked, their morphology, both macro and micro morphology. And so that's what people had to base their classifications on. And then the molecular era came in, and it just turned everything on its head, because it turns out that morphology in general just tends to be really unreliable when trying to understand relationships amongst the different groups of fungi.
In the case of Psilocybe, what other genera are the closest to Psilocybe?
So there's a number of them. Gymnopolis is fairly closely related. I actually have to refer back to my phylogeny to be able to tell you exactly. Let's see what else we have here. Cunearomyces, Hyphiloma, Stropharia is relatively closely related. Yeah, sorry, I can't think off the top of my head of what else. But so one thing is, all of the agarics that produce dark spores, they're all relatively closely related to each other. We know that, but that's a derived condition. And so Psilocybe, Stropharia, and Agaricus, and all of these taxa that have these dark spores, they're relatively closely related, as opposed to like Mycena or Tricholoma or these kinds of genera that have pale spores. Those are more distantly related in general.
And Bryn, so is psilocybin only found in the genus Psilocybe? So we tend to refer to, so people talk a lot about magic mushrooms. We have this like vernacular grouping of species in magic mushrooms, which basically means mushrooms with psychedelic compounds. But we know that Psilocybe and Amanita muscaria, so Psilocybe has dark spores and Amanita muscaria has white spores, so they're not as closely related based on what you're saying now. But Amanita muscaria is also considered a magic mushroom. But what is it that makes a mushroom magic? And in the case of the genus Psilocybe, what is it is it only in Psilocybe that we find psilocybin, basically?
Well, all mushrooms are magical to me. I'll start there. So let's start with Amanita muscaria. It has psychoactive properties. It has chemicals that interact with receptors in your brain and can alter your state of consciousness. But it's a completely different group of chemicals than what's found in Psilocybe. And the experience of their intoxication, if you will, is totally different. So those are not the same thing. And to use one term to refer to the two together is really grossly inaccurate, I would say. So, but that said, the chemical psilocybin, which is mostly associated with the genus Psilocybe, is found in a couple of other mushrooms outside of Psilocybe. And this has recently been a very interesting piece of the puzzle that has been essentially solved in the last couple of years. So it turns out that Paniolus is a genus that's very closely related to Psilocybe, and it's got a couple of species that contain this chemical as well. And it turns out that it's acquired the ability to manufacture this chemical horizontally. This is a phenomenon in evolution called horizontal gene transfer. So what happened was the genes responsible for the biosynthesis of psilocybin in psilocybe got moved over to the paneolus. And so horizontally,
How?
so instead of, we don't know that. We think what's happened. So a really interesting pattern, the sample size is quite low right now, but the really interesting pattern that emerged from this discovery was that species that do not share a common ancestor, like paneolus and psilocybe, but share the cluster, if they occur in the same habitat, the gene clusters that they contained seem to be more closely related than they are to the other clusters that make the same compound in other taxa. So even within psilocybe, we have taxa that are found on dung and taxa that are found on decaying wood, and they all produce psilocybin, and they all have a gene cluster that synthesizes this chemical. But it turns out if you look at the gene cluster that you find in paneolus, which occurs on dung, it looks more similar to the one on dung than the two gene clusters in psilocybe do to each other.
Wow.
That makes sense. So
yes, it does.
The implication is that what's happened is the gene cluster has been transferred via the habitat across organisms. And that's really intriguing. And probably the most likely scenario for how this occurs is through hyphal contact. So the fungi are competing in this environment, and this is especially true in dung habitats because they're very short-lived. So they're competing a lot for the resources that are available, and they are going to be interacting and battling each other and having this physical contact. And so we think most likely what's happened is parts of DNA from one organism have moved over into the other during these interactions.
So is that equivalent to hybridization?
No, it's not hybridization. This is different. This is just small sections of the genome which get moved out of one organism into another. It was thought that this phenomenon was quite rare. It was best known in bacteria. But now that we've been able to interrogate genomes of many different species, we've discovered that, and this is not only in fungi, but in other organisms, particularly plants, horizontal gene transfer is a lot more common than we thought. So it looks like our boundaries between organisms that we try to classify in our world around us are more fluid than we once thought.
That's incredible. I'd never heard of that.
It is quite incredible. Yeah, it is quite incredible.
So paneolus has psilocybin. I know just from literature that the genus Inocybe has also some species that have psilocybin. But I'm really interested to know whether every species that contains psilocybin, are they all cap and stipe fungi? Do they all have a pileus and a stipe? Or can you find, I don't know, a coralloid form with psilocybin or psilocin? I don't know them in English.
Yeah, I can explain the chemistry a bit if you want, but the short answer is, at the moment, we don't know of other taxa other than pileate, stipitate, things with a cap and stalk that contain the chemical, although there's at least anecdotal evidence and some preliminary chemical evidence that there's a lichenized basidiomycete, so a relative of the wax cap fungi, hygrospes, that may potentially contain psilocybin, and that doesn't that doesn't have a cap and stalk. It's lichenized. Looks weird. And then there's at least one truffle form that has evolved within psilocybe. So the truffle morphology, truffles, yeah. And I use that in a loose sense. I don't mean the culinary truffle, which is the genus tuber. I mean reproductive structures, mushrooms that occur underground, and we call that.
So hypogeous sporomes.
Yeah. And those typically require animals to dig them up and vector their spores typically.
So you're saying, is this anecdotal evidence or molecular spores? Here, Paul Stamets is saying Massospora has psilocin. Werero was commented also from Kane, who's in Melbourne. But can you explain a bit what's going on there with these genera?
The Werero is what I was referring to in this case. That's been transferred to Psilocybe now because it's been shown that it's nested within Psilocybe. So that's a case of the truffle form that's evolved within Psilocybe. At present, there's no chemical analysis to confirm that it contains psilocybin, but if you go on Reddit, you can certainly see that it looks like it probably does.
Okay.
And then Paul mentions Massospora. This is a very interesting case of psilocybin, probable psilocybin production, although I only say probable because it turns out that the genes required for producing psilocybin have not been identified yet. And so we don't quite know exactly what's going on. But Massospora is a obligate parasite of cicadas.
Oh, wow.
It's thought to be manipulating the behavior of the cicadas through its production of psilocybin and some other chemicals. So that's a very interesting scenario.
Is this the paper? I was reading some of your work earlier. Is this the work that you are one of the authors on?
Not the Massospora, no. We published an article looking at the gene cluster in some of these other species. So we confirmed some of the work that came out before ours on the gene cluster that is responsible for the synthesis of psilocybin in Psilocybe. And we were looking at Psilocybe cyanescens, the species cyanescens, which, well, I probably shouldn't say, is easily accessed in the UK.
Okay.
So it was abundant.
You can say all available. That. For a night with Julie. You can say whatever you.
It was first described from landscaping at Kew Gardens. So when I was at Kew, it was fairly easy to acquire some of this material for our studies.
Okay.
So we were looking at that, but then we also started to look at some of these other genera, like Inocybe, like Pluteus salicinus, another independent origin of psilocybin production, and that's been verified chemically. And we found something.
So there's a Pluteus, a Paniolus, Inocybe that have psilocybin.
Yeah. And
apart from Gymnopilus, Gymnopilus and others that we know. Okay.
Yeah. And excuse me. We found something really interesting in the Inocybe. So we found the same cluster in gene cluster. I should probably back up and explain a little bit that biosynthetic gene clusters and biosynthetic genes in fungi tend to be clustered, which means that all the genes necessary to complete a biosynthetic pathway are physically close to each other within the genome. That's a common pattern in fungi. So when I'm talking about gene clusters, this is what I mean. That we have a cluster of genes, all of which perform a step in the synthesis of psilocybin from the amino acid tryptophan.
Wow.
So that makes it easier for us to go in without any prior knowledge and kind of scan through genomes looking for candidate gene clusters, because we know they should all be next to each other, generally speaking. So when we started this out, we went in and knew the chemical transformations that were required to convert tryptophan into psilocybin. We knew what the chemistry was, which allowed us then to go in and try to find genes that would perform those functions and that were close to each other in the genome. And when we did this with our Psilocybe cyanescens genome, we found 3 gene clusters, all of which looked like they were potentially capable of producing psilocybin from tryptophan. Now, only one of these has been confirmed biochemically to do this. The other 2 that we've identified may perform the same or similar biosynthetic roles in the fungus. So there's the potential that there are still other gene clusters that we haven't looked into deeply enough to know whether or not they're making psilocybin also, or psilocybin-like chemicals.
And are those 3 known gene clusters close to each other?
No, they're scattered throughout.
Okay. That's super interesting. Thank you for that. There are a few questions that I've prepared, and there are a few questions that we've received from people during the day. But the first thing I want to ask is, do we know how, for example, how old psilocybes are? So how long ago did the genus Psilocybe, as we know it today, appear on Earth, the genus?
Well, we kind of know-ish. So one problem with trying to put absolute dates on fungi is that they leave a very poor fossil record, and so unless you have physical evidence.
There's one. We were together. We met one together with Maria Alice, who I think is connected in Florianopolis. We held in our hands the supposed oldest fossilized agaric on Earth, right? That's for a whole other world.
I'm personally skeptical of that fossil, but nonetheless, they leave a poor fossil record. So we don't have any fossilized psilocybes. And even when we do have fossilized mushrooms, they tend to be relatively young. And so they don't give us a very good sort of like minimum age. But they also, because all we have to go on is morphology, and we know that morphology is not terribly reliable when it comes to classification, it's just really hard to place confidence in the position of those fossils on phylogenies today. So that's all the caveat leading up to what I'm going to tell you now, which is I actually was doing a little bit of this work to try to understand, how old these might be. And I'm going to turn my screen around here, if that's okay. Okay, so what you're looking at is the family tree of Psilocybe and some closer relatives. And
okay, wait, sorry, Bryn. Before you go into that, I'm going to tell people we are going to post this on Bryn's blog. We will send out the link. So you'll have to explain it because it's really invisible. And also, some people, Paul, I can send this to you. I know you're super interested in this with Bryn's permission. So before we go into it, just please explain it all, Bryn, because we can't see a single thing. Okay.
Okay. Oh, sorry. Can you see it at all? Is this worth even showing?
Go further away, and we'll be able to see it.
Further away.
Yeah. And then tap on the screen to focus the tree. No, I can't see the tree, but you're gonna have to explain it. You're gonna have to improvise.
Okay.
Go for it.
Well, I'm just gonna turn the camera around and explain. So, if people are interested, they can look later. We'll share this somehow. But what I have is a tree that is based on the most recent molecular phylogeny of Psilocybe, which was published by Ramirez Cruz et al. In 2013. Which to me that sounds recent, but I know that a 2013 paper is now like ancient history in science because it's moving so fast.
Well, because we have a lot of older work that we refer to.
Yeah, so this is the best I think that we have to work with right now. And
sorry, can you repeat the name of it?
Ramirez Cruz is the first
Ramirez Cruz. Okay, yeah. 2013. Perfect. Thank you.
And so what I did is I took their tree and then I converted the branch lengths on that tree into time. And I did that using a statistical method called penalized likelihood. And you don't need to worry about the details here.
Okay.
And what I did is I set the root node to 70 million years. And that's based on a recent sort of meta-analysis of divergence times within the Basidiomycota that was published just last year.
Okay.
Another paper by Hay et al. So I did this.
By who? Sorry, because they're writing them in.
Hay et al. H-E.
Hey. H-E. Okay.
Yeah. Okay, so this is all very hand-wavy. I can't be absolutely certain that these dates are correct. But from this analysis, the genus Psilocybe, the ones that contain psilocybin, evolved 28.3 million years ago.
28.3 million.
Yeah.
Years
now there's
ago.
going to be huge error bars on this, and I didn't do like a super rigorous analysis. So,
okay.
I would say
give or take.
I would say they evolved 28.3 million years ago, give or take 28.3 million years. I can't have a ton of confidence in this.
Yeah.
That said, this is a fairly reasonable age. If you look across what other ages are for similar groups, this is not an unreasonable number. And I think the take home would be that we're measuring the age of this group in millions of years, not in thousands of years.
Yeah.
Or hundreds of years. Or hundreds of millions of years. It's in the millions of years category. They certainly predate humans.
Oh, they predate humans.
By many millions of years, yeah.
So we can say they've always been around.
Psilocybin was on Earth before humans evolved.
Were they on Earth? Okay, we're going to have to compare. Somebody go back on internet. Anybody who's watching, tell us what was around 28.3 million years ago, because I'm like, anyway. What, also tell us what city and country you're connecting from. This is, okay, sorry.
Yeah, that's fine.
So that means that, and what species of the genus Psilocybe is the oldest?
Well, we don't really know that yet because there are over 300 described species of Psilocybe, but we have not included all of them in a molecular phylogeny. This is a thing we need to do, right?
We need to get all 300 species, put them in the phylogeny, and then clock it molecularly, and see the ancestries between species of the same genus.
If we want to answer that question, we would need to do that. Yeah.
Okay.
So right now,
is that doable?
Oh yeah.
Okay.
Yeah. It's a matter of getting the material to do the work. And as you know, and maybe many of your viewers know, it's hard to get these species because they're ephemeral and they occur unpredictably. So in mycology, we really suffer when it comes to trying to sample taxa for our studies a lot more than people that work on other groups like birds.
Yeah, of course. Okay, so we don't know what the first Psilocybe species was, but let's ask ourselves, for example, emblematic species. Can you give us some times of your tree and your clocking? We know all the bars, error bars, but go for it. Tell us species and years, Bryn. And they're not my viewers; they're our viewers. Have to say.
Okay. So there's two major groups within Psilocybe. There's the group that contains Psilocybe cubensis and Psilocybe cyanescens. These are probably two of the best known species. And then there's another group, and that contains things like Psilocybe mexicana, Psilocybe zapoticorum. Other species that might be familiar to people. So those are two big divisions within the genus. And I should say, going back to the conversation we were having about this psilocybin biosynthesis, we don't yet have any genomic information from that other clade that includes mexicana and zapoticorum, et cetera, et cetera.
There's never been a whole genome extracted from the clade. Is it a clade?
Yeah, we'd call that a clade. Yeah.
It's a whole clade that has not, so there are two big clades. One has had its genome sequenced and the other hasn't. That's incredible because we're talking about, really top-notch, pharmaceutical, industrial development, and we don't know the genome of a whole clade. I'm just putting that out there.
Well, we don't know the genomes of many clades, I guess, but, it's difficult to do this work. partly because, accessing the material is just difficult in and of itself.
Yeah. But I'm talking about within the psychedelic medicine industry.
Yeah, but it's still not legal, right? And so in most places. And so it's still difficult for people like me to be able to acquire the things we need to acquire to do this work, because we would need licenses from the Drug Enforcement Agency just to even handle these things, right? So my lab is doing this. My lab has, we're collaborating with people that have a DEA license, so we can do some of this work. And we are currently generating, we're not currently doing it because we're all stuck at home, but we are working on getting a genome from that other clade currently. Now, the expectation is that the cluster is going to look the same, that it's probably been inherited from the common ancestor of the group. Yeah. But until we look and are certain of that, we can't really say. Yeah. So to get back to your original question.
Thank you for bearing with me. This happens a lot. Okay.
So Psilocybe cubensis, it looks like, so to talk about ages, you have to talk about stem ages. So the stem is the point at which that lineage first emerged. Okay, and then the lineage can diversify into multiple species.
So in a tree, it's the horizontal line. It's not the vertical lines.
Yeah, so it's the long line that leads up to a cluster of species. Yeah, that's the stem. So the stem age for Psilocybe cubensis is approximately 18 million years. And then Psilocybe cubensis diverged from subcubensis, or vice versa, approximately 5 million years ago.
5 million years ago. It's a recent. Wow.
Still older than humans.
Sorry?
Still older than humans.
It's still older than humans. Yeah, but now, okay. It's still older than humans. But so what about cyanescens?
So cyanescens.
Sorry, wait. Have a sip. I'm going to talk, so you can have a sip of your wine, if you want.
Yeah. Okay. Go for it.
Okay. While you look for cyanescens, no. I'm totally blown away by this. This is all really, really amazing information. So 5 million years for cubensis from a stem that you said how many years?
For cubensis or cyanescens?
Cubensis, the stem.
Cubensis is approximately 18 million years.
18, and cubensis 5. Okay,
and then diversified more recently, yeah.
And what about cyanescens? Do you have it?
Yeah, so cyanescens, the stem age is about 10 million years. And then it also diversified approximately the same time as cubensis, around 5 million years-ish.
What about the other known psychoactive, Stuntii and Alani, and there's the Tampanensis, and what about those?
So stem clade for Stuntii. So Stuntii shares a most recent common ancestor with Hispanica pelliculosa, an unidentified species, and Cerulipes, and that whole clade evolved around 10 million years ago. Stuntii diverged relatively recently from Hispanica, past couple of million years probably.
Wow. So we're talking about ish. Okay. I just want to ask everyone. Okay, we have a question, but before I ask that, were Psilocybe originally saprobic, or saprobes?
Yeah, almost certainly.
Did they move into dung, or the other way around, from dung to detritus?
It's likely that they colonized dung habitats multiple times.
What does that mean?
So it's likely that the ancestor was a wood decayer, and then species diverged and colonized dung from them more than once.
More than once. So it went wood, dung, wood, dung, wood, dung, wood.
Yeah, so that's the kind of thing we would be able to know and answer if we had a complete comprehensive phylogeny of the family. Of the genus. Until we have that, we can't really say for sure what the pattern is. But most likely, because most of these are not dung inhabiting, most likely the dung habitat was colonized, is a derived condition.
Wow. So is it can one does the tool exist to track that same species back into those habits?
I mean, we can infer. Is that what you mean, like an ancestral condition?
Yeah. You can. Okay. Yeah, people can infer that.
We have statistical tools that we can apply to infer what the ancestor was like.
Yeah. I just want to tell everybody that I'm outside in the pouring rain. Can you go deeper into that? I'm just going to get my raincoat and I'll be right back. Give me 1 minute to get my raincoat.
So we're talking about ancestral conditions. So we can sort of infer what the ancestral condition was based on the distribution of known traits on the terminals of the phylogeny. So if everything except one was a wood inhabiting fungus, then we probably would infer that the ancestor was also wood inhabiting, just because there are more wood inhabiting now. What's the likelihood that we'd see a change more than once? So that would be like applying maximum parsimony. So the simplest pattern is the correct pattern. That may not be true.
Yeah.
And there are other idiosyncrasies of phylogenies that can help inform what some of these ancestral conditions are, namely branch lengths. That can be really informative.
So we have some questions from people of our viewers. I want to just confirm that if Paul, if you're connected, I'm actually really, really proud to wear a raincoat that was donated to the foundation by a friend of Paul's who I met through Paul, which is Bruce Lingerie. So I'm just putting that out there because he's never looked for recognition, but we're dry in the field thanks to a philanthropist friend of Paul's who I met through Paul's family. So thank you very much for that. There was a question before from Guerrilla Mycology, my good friend Antonio, who's the best, probably one of the best mushroom accounts on Instagram, Mushroom Gorilla, who asked about the do we know whether Psilocybe co-evolved with ruminants?
We don't know that. Co-evolved, probably not. So co-evolution requires reciprocal change.
Yeah.
And it may be possible that we've seen change in the ruminants in response to change in Psilocybe, but I suspect that's unlikely. So it's more likely that Psilocybe evolved adaptations to make use of ruminants as vectors, but that the ruminants have not responded in any way.
Do we know of effects of psilocybin on animals other than humans?
A little bit. I mean, there used to be research on this before Richard Nixon declared it a scourge of the earth.
It's all about Nixon. That's actually in the movie too. Yeah.
And so people were giving psilocybin to various animals, and it definitely affects other animals. And we looked into what was known about its effects on invertebrates, because the assumption is that a bioactive chemical like this evolved as a defense against something, and mushrooms are often eaten by invertebrates of various types. So
we'll find a fly or something, or
so people injected psilocybin into what was it now? Spiders.
Wow.
And they made crazy webs. And then LSD has similar activities.
Made crazy webs? So their webs were altered when they were injected with psilocybin?
Yeah.
No.
Yeah.
Really?
Yeah, really. Yeah.
Oh my God! Can you send that the reference around? Well, afterwards, later. We'll do that later.
I'm gonna double check my memory of that. LSD has similar activity to psilocybin. It's a bit more promiscuous in the brain than psilocybin. That's one of the advantages that the medical community has identified in psilocybin, is that it's got a very narrow activity in the brain. So it's easier to work with. It's easier to justify working with.
Psilocybin. I was moderating a panel with Dave Nichols at the Telluride Mushroom Festival last year, and he was explaining that, and it's the same, I think, with DMT and with other with MDMA, that the routes change, and the complexity of the routes.
Yeah. So I was just rereading my paper just to make sure I was right about the spider thing, and that is true. Yeah, spiders were given psilocybin and they made funny webs. And then, Drosophila flies have been given LSD and then put in these weird flying arenas where they they spin contrasting colors around, and the flies have to orient themselves. This sounds kind of trippy to begin with, and then you give them LSD and they showed modified behavior. So that suggests that these chemicals that interact with serotonin receptors in mammals also interact with serotonin receptors that have neurological function in invertebrates. Now, we don't really know. We can't say for sure because there's very little data on this right now, but it seems likely that that's the case. However, I think it's really worth mentioning that a very recent study came out, which is totally fascinating from my perspective, that elucidated for the first time the chemical basis of the blue staining color reaction in psilocybin-containing mushrooms. So you may not know this, Giuliana, but psilocybin-containing mushrooms.
Tell me, enlighten me here, Bryn.
They almost invariably turn blue or deep blue when they're handled or in age. And that turns out to be a reaction that is catalyzed by an enzyme. And what it does is it ends up dimerizing. So it ends up taking two psilocin molecules and putting them together. And when that happens, you get a chromophore, which then reflects blue light.
Hold on. So, go on. Do it again. Say it again.
So it takes two molecules of psilocin, puts them together, and when this happens, you get what's called a chromophore.
Yeah.
And the energy state of the chromophore causes blue light to be reflected. And it's the same principle in indigo dye, actually, which uses chemicals that are not too different from psilocin, excuse me.
And so yeah, please.
So what's really interesting, so that is the chemical basis of the blue color. Now you might ask why the fungus is doing this. What is the point of putting things together? Well, it turns out that this is all surmised, but that these long chains of psilocin function similarly to chemicals you find in plants that are anti-herbivory chemicals. So these chains of psilocin can release free radicals in the guts of the insects that consume them, which will then cause lesions in their gut lining, which will then...
Wow.
And so it's possible that psilocybin evolved not because of its direct activity in the brain, but because it's used as a unit to create these toxic substances that cause lesions in insect guts.
Shit. When was that sort of inferred?
This is a brand new paper. This just came out in the last couple of months or late last year.
So, what are you doing with psilocybe? Why do you know all this? What's going on with you and psilocybe? What's your thing with psilocybe, Bryn?
Well, I got interested in this way back in 2014 when I realized that nobody knew what the chemical basis, the biosynthetic basis of psilocybin was. And so I set out because psilocybe cyanescens was so readily available to me, and genomic technology really had just like become a thing, and it was affordable and accessible. I sort of saw this as a low-hanging fruit. And so I started to investigate this without funding. And throughout this whole process, I haven't had any funding, which is why we've been really slow relative to these other groups. So we've been scooped about 3 or 4 times now.
What does scooped mean?
Oh, somebody publishes what you were going to publish before you do.
Oh, okay.
It's called scooping.
Oh, okay. Oh, I know it.
So,
yeah. Maybe, okay.
In academic publishing, you need to be the first one to get the credit. Is basically how it works out.
So what are you doing with psilocybe? What's going on?
Well, so I sort of already explained what we were doing. We're trying to understand the biosynthetic basis of psilocybin production in cyanescens, and then we were just about ready to publish it when the first paper came out, and that was a blow. So what we did is we went back and we looked at what questions were unanswered yet, and one of them was, well, do these independent origins of psilocybin production, what is the biosynthetic basis of those? Is it the same, or did they evolve independently? And in most cases that we looked at, and there aren't many of them, of course, it looks like it mostly achieved through horizontal gene transfer, like we had discussed.
Horizontal.
Yeah, except in one case, and this is quite interesting. So in the genus Inocybe, we have an independent origin of psilocybin production that's been confirmed chemically by previous researchers and again by our group.
Yeah.
That species does not contain the same gene cluster as all the other psilocybin species that we know about.
So one species.
We've only looked at one species so far. We could expect probably that all of the species of Inocybe that produce psilocybin share the same gene cluster. That would be a parsimonious expectation, but we don't know until we sequence them. And what's really interesting is we found another cluster in the Inocybe genome that contains genes that look like they would perform the same 4 steps in psilocybin biosynthesis, but they evolved independently. So it's a completely novel origin of psilocybin biosynthesis in Inocybe, we think. So we're currently trying to confirm this discovery.
Yeah. Sorry, Alia Muhic, our friend and colleague, I dare say, is connected. Alia, I said shit. He was laughing because I said shit. I'm saying shit again. So what you're saying, Bryn, is that psilocybin was produced in an evolutionary time for some species, for some genera, but there's this one genera that we know of that produced psilocybin in a parallel route. Is that what you're saying?
That's right. So we call this convergent evolution.
That's what. So Doug Beer and another friend, he says, "Is that an example?" Hi, Doug. We love you, Doug. A massive hug to you, Doug. I know it's been a hard week for you. Doug Beerand, amazing author, has had a hard week. I'm sending you much love. Love you, Doug. He's asking, is that an example of convergent evolution? And you were just saying, yes, it is.
Yeah. Yeah, and that's really strong evidence for the adaptive role of something. So if something has an adaptive advantage, and it's likely to emerge more than once if it's possible to do that. Or
for
thinking
example,
about it
going into a truffle. Yeah. Like going into a truffle
emerge more than once independently, then it's likely adaptive.
Wow. Shit.
Yeah, it's quite cool.
Doing it again, sorry.
I mean, it's really, I think this particular story is, I just think it's such an amazing story because it illustrates not only the power of sort of non-vertically transmitted inheritance to give rise to adaptation in organisms, but at the same time, it illustrates the power of convergent evolution, like the power of selection to create things more than once. I mean, it's really just kind of an amazing story.
Eyes, or right?
Yeah, eyes are like the classic convergent evolution, or wings. Yeah, yep.
It's really, really incredible. That, I mean, because this, I mean, for fungi, it's so novel, it's so new that we're experimenting this for the first few times in a whole kingdom, right?
Yeah, right. For sure. We don't, and I should say, we don't actually know where the psilocybin biosynthetic cluster originated. And I should also say, so we don't know where it came from to begin with, that when you start looking at similar clusters in fungi, that it looks like the ability to produce chemicals related to psilocybin is quite widespread within the kingdom, far more so than we thought. And so, and in fact, there was a very recent study that reported the heterologous expression of psilocybin biosynthesis in yeast. So they were able to modify yeast to produce psilocybin. Turns out that they were able to do this not by taking just the psilocybin biosynthetic cluster itself, but by adding to it another gene from the genome that performs a similar, identical function to one of the genes in the cluster. That suggests that this ability to produce chemicals like this is ancient and widespread. So
wow.
we are fascinated with psilocybin because it's something that we know about, but there's potentially other chemicals out there that are possibly psychoactive, but may also have as good or better therapeutic properties than psilocybin in mushrooms that we just simply don't know about yet.
It's incredible. And so, there have been like a lot of questions, and I haven't, been able to get through all of them quickly and everything. So there's a question that asks whether humans followed maybe. So if our evolution, you said, for example, it's probable, when we had the question if ruminants co-evolved with fungi, and you said it was probable that ruminants evolved sort of conditioned by fungi. Do you think that's the case for us as well, for humans?
Well, I'm a mycologist. Let me just put it, my expertise is about fungi and fungal diversity and evolution. I'm very skeptical of the idea that human brains evolved in response to consuming psychoactive substances. Who am I to say, really? But I think ultimately the problem with these stories is they're 100% untestable. So, maybe the possibility is there, but will we ever know? We won't ever know, unfortunately. They're fun to think about.
Example, if we go to the exact, for example, case of the hominids, for example, in the movie Fantastic Fungi, which I hope you've seen. Have you seen Fantastic Fungi?
I have.
You have. You have.
Yeah.
Of course.
We've got a great little art house theater here in Salt Lake City that had it, and I saw it like one of the snaps that it was showing.
So cool. Okay, so in the movie, there's a scene where there's this, there's a recreation of our ancestors. So not even, I think it's before Homo sapiens, right? I can't remember exactly. It's based on, something published that suggests that primate or human, apes were eating psilocybe because they were psychoactive before, our brain had evolved. And what you're saying, that our brain, changed with them. Can you explain where that comes from? Do you know the citation? Do you know the work?
I'm familiar with the stoned ape hypothesis, yes.
Thank you. Yeah, that was it. All right. So what's the stoned ape hypothesis?
Well, I do not study human evolution. I do not study neuroscience. I am not an expert that should be weighing in on these topics whatsoever.
But you in your bio, it says that you worked in but you have some neuroscientific interests.
That's kind of where this all started, to be perfectly honest.
Okay, so why? What do you mean?
Well, I'm fascinated. I continue to be fascinated by altered states of consciousness. I think they inform us a lot about reality. I just heard a quote recently about art that I think probably applies to altered states of consciousness as well, which is, let me see if I can recreate it. It's the lie that tells us the truth about reality.
Art is the lie that tells us the truth about reality. Okay, listen. We're going to talk about this right now, about that. Instagram is cutting us off in 2 seconds. That it's not it just so happens.
Well, it's just not a feature you could use to classify things by, but it is a feature in this case that you can use to distinguish them.
Shit. Okay, that's a good one. Okay. That's really beautifully said, have to say. Okay, so what can we expect? Can we expect to find other genera of fungi, of mushrooms, and fungi, not only mushrooms, that produce psilocybin?
I definitely think it's possible. There are anecdotes of other psychoactive mushrooms that are consumed around the world by various cultures that have never been investigated. The chemical basis is maybe the chemical basis was investigated a little bit, but hasn't been thoroughly investigated. So I think it's possible we'll find other things.
Can you give us examples of those? For example, what uses, what anecdotal notes are there of use of mushrooms that haven't been investigated? There's a lichen in Ecuador, they say.
Yeah, right, in the Amazon basin.
The one you were mentioning. But, I mean, my professor, my number one main mushroom man, Don Feaster, thinks that's complete bullshit. But
yeah,
I have to say, okay, so what do you think?
Well, I need to connect with a colleague of mine who has offered to send me material to sequence. There has been a preliminary chemical analysis which could not refute that there was psilocybin present. But the problem with a lot of chemical analyses is
it can't refute it, but it can't prove it.
Yeah, so chemical analysis is very tricky. And one problem is you need to have a chemical standard that you can use to compare your unknown to, right? To confirm that what you think it is actually the thing that it is. And that chemical standard is regulated by the DEA. and it has been up until recently very difficult to obtain. And so the chemical analysis that's been done previously couldn't confirm that the chemical they were seeing was truly psilocybin or something that looked similar. So this study needs to be followed up for sure. I think it's definitely possible.
So just so that people can understand, and me, because I mean, these Myco Viral Nights, they're pretty, a lot of my doubts too, but how do you test a mushroom? Do you collect it in the field, you have a dried sample, I don't know, and then you take it to a machine that grinds it, I don't know, and then like juices it, and then says, and then you reflect light on it, or what's going on?
Well, it depends on what kind of analysis you're talking about. So for chemical analysis, pure like trying to identify the presence of psilocybin, you would take a sample, preferably fresh sample, acetone.
Water. Yeah, water.
Yeah, for sure. Any of these are solvents, so anything that a chemical will dissolve in is a solvent. So you would dissolve, you would grind up your tissue in a solvent, and you take that solvent and you'd inject it into a machine. And there's a couple of different machines that can separate chemicals by their properties. So by their polarity and by their mass and by the parts that they break into if you bombard them with electrons.
Wow, yeah.
And they break into sort of signature parts that you can then detect based on the mass.
Right. You can you can unequivocally measure that.
Yeah, so you break these things apart, and then you try to infer from the parts that you see. You try to put the puzzle back together, and you try to put Humpty Dumpty back together, and see if it really is Humpty Dumpty. And the challenge there is you can't actually see the little parts themselves directly. You have to infer it, right? And the best way you infer things is if you have a reference that you know is true. And that's been a real challenge because of the regulations, the licensing.
Yeah. Wow. So there's a massive industry, incipiente we say. How do you say incipiente?
Incipient.
There's an incipient, almost consolidated industry that is producing psilocybin, or is extracting psilocybin from psilocybe for pharmaceutical purposes, for therapeutical ultimately, because it's medicine. And I'd really love, before we start talking today, we talk a bit about our perceptions and what's going on with the medicinal use of psilocybe. But they are, there's no regulation, there's a lot of funding, there's a massive demand. And how do you see the genetics of the genus in comparison to that, or in parallel to that? What was the science versus the hype? So when you have Silicon Valley, you'd expect. So when they were producing computers and phones and smartphones, you've got to have, technology and engineering and electricity and all that going on in parallel to the boom, to the demand and the offer. What's going on with psilocybin?
Well, so I mean, I think this is going to be difficult.
You gotta do it. We say in Chile, you gotta wet your butt to cross the river. You just gotta wet the butt. It's like it's fine.
Well, I'll try to do this without wetting my butt.
You're not representing an institution.
So what I can say, what I know is the technology exists to be able to create genetically modified organisms that can produce chemicals that are of interest in large quantities very cheaply. So you could take a gene cluster from a mushroom or any other organism in theory, and insert it into another organism and get that organism to make that stuff.
GMO. So that's what GMO is, right?
It's a type of GMO, yeah. And so this has been done recently because psilocybin is chemically actually quite difficult to create synthetically in a lab, and that has to do with a particular feature of it that's just very difficult. Chemists can't do it very well. Enzymes that the mushrooms have evolved do it very well. So the holy grail here is to get the enzyme, to take the enzyme and then be able to control it under our laboratory controlled conditions.
Which is what's happened with LSD.
Well, there's no enzyme that produces LSD at the moment.
Right.
It's all simple.
But
yeah.
No, but apart from the enzyme, it's the ability for a laboratory to, to create it.
Yeah, because these mushrooms that produce it naturally, they're doing it under their, they have their own agenda. And we can't tell it what to do. Like, they don't listen to us. They're like 3-year-old children. They're never going to do what you want them to do. And so if you want to get
13-year-olds don't either.
Or any age child, really, right? Yeah. So if you really want to get this substance, you need to have complete control over it. And the way that biotechnology has evolved is being able to utilize these organisms, like yeast especially, but there's some other organisms, to be able to produce these chemicals on demand. And that's been done recently. So I think the bridge with Silicon Valley is actually the biotechnology that allows us to produce large quantities of psilocybin very cheaply. And that breakthrough has been achieved. However, I would say another recent discovery in the mushrooms is that psilocybin does not act independently in the mushrooms. Its bioactivity, in fact, is synergistic with other chemicals that the mushrooms are making.
Which is what you talked about with the yeast, right? It wasn't the gene cluster alone wasn't put in to produce psilocybin, but you had to accompany it with other genes. Okay, so I'm getting it. Sorry.
There's an analogy there, but this is what I'm saying is a little different. So there are other chemicals that the mushrooms are producing that might act synergistically with psilocybin when a person or other animal consumes it, right? That actually produces the phenotype of consuming the mushroom that you won't achieve with a single molecule in a pill, right?
Right.
And it turns out that there's a chemical that's manufactured in these mushrooms that inhibits this enzyme that would detoxify psilocybin in the gut. Monoamine oxidase inhibitors. So it functions, magic mushrooms are kind of like miniature ayahuascas. If you know ayahuasca, the concoction.
Where you need two plants. You need rainha and yabe.
So the two chemicals are DMT and a beta carbolate.
Love ayahuasca.
Monoamine oxidase inhibitor. And it turns out that magic mushrooms have the same two types of chemicals. So it's quite likely that when you consume a whole mushroom, if you were to do that, you would have a very different experience than if you were to take the equivalent amount of psilocybin alone. And that's because of
we had that question. So we had a question from Daniel Puig 8. Daniel, I don't know where you are writing from, I suppose Chile. So what you're saying basically is that taking the mushroom is different than taking lab-produced psilocybe. That was his question. Is there a difference? Definitely.
I mean, I've never taken pure psilocybin produced by a lab, but I'm no doubt there would be a different experience.
Right. So they were. A lot of people are really interested. I'm having a visit from Moths. Is there MAO in psilocybe? Yeah. Oh my God. Everybody's okay. So there's MAO in psilocybe.
Yes, MAO inhibitors. Yeah.
In this, okay. Is there MAO in IMAO in Gymnopolis that we know of?
It's not known.
It's not known, so the laughing
it's only been shown in one species at present. But now that we know it exists, we can start looking in the other genomes.
Okay, so they're asking, is there a paper for that? There's a question from Australia.
There is, yep.
There is a paper for that. Okay, so Kane, you can write to me or to the fund. You've got my email, I think, and we can get that paper on. Or if you know the name of the paper, Bryn, we can give just give the name and year. Do you know it?
I can't remember.
Don't look for it. Don't look for it. We're going to give a list. The people at the Fungi Foundation are taping this, recording it. This is going to be available, and it's going to be translated. It's going to be subtitled for internet. I just want to do a shout out. There's an account called Psychodemia _CL, and that's two dear friends of mine, Mario and America. And I just want to say, America is the translator for the foundation. She is a simultaneous translator, and she translates. She has translated, like revised the whole movie, Fantastic Fungi, and everything we do. I just want to say to America, we love you. She is a part of our foundation. And thank you. She's going to translate all of this with the foundation, and we're going to make it available. Bryn, more about what you're doing now. We're going to wrap up soon because we could go on surely. Yeah. But I just want to ask you about what you think is going to happen now in the future with everything that's happening around magic mushrooms and this whole industry and getting to finally clock the genus and the whole. How is the molecular era and the development of psilocybin medicine working together or not, or how are they like into each other?
Boy, I really don't know.
What do you think?
I think it's going to be, so there's a lot of, it's very trendy to be microdosing right now. And of course, antidepressants that have been developed by the pharmaceutical industry over the last decades have proven to be pretty much total failures. They're very, very ineffective over the long term. And I think the real promise in psilocybin is that it seems to have real effects for a long period of time after the application. And so unlike any other antidepressant that's ever been examined. So I think people are very excited about this, and for good reason. But
the trials, I'm sorry to interrupt you, but the trials are with synthesized psilocybin and not with the accompanying compounds of the mushroom. That has to be said.
Yeah, yes, fair enough. But I think when you're talking about trying to work within this highly sanitized medical environment, you have to work. You have to reduce complexity. You have to reduce complexity because you have to be able to explain things mechanistically. Otherwise, it's no good than just telling a story. And I think there's real value in that approach. In this case, I think it's going to be super difficult to reconcile the sanitized medical approach to health with what seems to be something that is more on the spiritual side. So it seems like the medical benefits are achieved not by standard medical applications, right, but by the polar opposite of that, by inducing religious-like experiences. And I don't know what the future is for reconciling these two areas. I hope that there is some way to reconcile them for humanity, but I have no idea. I'm just a quaint mycologist.
But so basically, this is what when I was moderating the panel with Dave Nichols at the Telluride Mushroom Festival 2019, there were a lot of questions about microdosing from the public and people, and we've had dozens of questions about that. changes and betters, mejoras, I don't know how to say it, improvements, I don't know, without too many bad, effects, and through microdosing, you don't achieve that. And so the full potential of the medicinal effect of psilocybin is through a therapeutic dose and not microdosing. It seems to me that that's what you're saying too.
That's my understanding. Yeah. I mean, again, I'm not a neuroscientist. I'm not a psychiatrist. I have no authority whatsoever.
Yeah.
But it seems like, understanding the mechanism of action of psilocybin and knowing how antidepressants developed by the pharmaceutical industry work, my very limited understanding of it is they work similarly. So, microdosing being similar to taking antidepressants every day is probably going to be equally ineffective. I don't know.
Right.
But
yeah.
It seems to me that makes sense.
It does make sense. I mean, it definitely makes sense. So there's a question, I don't know if you know about this. What is known about woodlover paralysis? Do you know what that is? I have heard of it, but I don't know if you have, Bryn.
I've never heard of it. What is it?
Okay, so it's basically there's with a certain dosage of psilocybin, there's a moment where things stop in beauty for a long time, and then out.
I don't know. I think I would say if you're interested,
part of the measurement.
If you're interested in the neuroscience perspective on the psychedelic state induced by psilocybin, I would be reading Robin Carhart Harris. I think he's pioneering our understanding of the effects of psilocybin on consciousness in the brain. So I would highly recommend reading Robin Carhart Harris.
Can you write it in the comments here right now, please? So that's super cool because we're all, reading Michael Pollan and we've read, some of the things, but that's super valuable that you put that out there. I want to say hello to the American Construction League that has joined us. That's Nico and Matt and Martita from New York. They are heroes in the COVID-19 pandemic in New York, which is the worst place. They have for weeks been collecting masks and gloves and all sorts of protective wear from restaurants, tattoo salons, veterinarians, and other places, and donating it to hospitals in New York. So big shout out to the American Construction League for their philanthropy for weeks. And I love you, Nico. Anyway, so now we're getting a lot of questions about the dosage. We don't know that. We're not going to answer that, right, Bryn? We don't know, if it's best to take there. We'll leave that to, whoever wants to take that on. What else would you like to tell us about psilocybe in the molecular era? And then we're going to do a salut, and we're going to do our traditional out that you already know, where you are going to say goodbye and close our session.
Okay.
Okay.
I think we're just on the verge of having a really thorough understanding of these organisms and the biology behind how they, why they make these things and how they make them. We now know a lot about how they make them. We know very little, if nothing, about why they make them. So I think the next frontier with psilocybe, from my perspective, is understanding truly, why do they produce these chemicals? Why, what is the function in nature?
Yeah.
And that's where my research is going to be heading in the future.
Although you've already mentioned this, I mean, for me, it's really incredible that psilocybin wounds the tract. So the blue producing compounds, not psilocybin, sorry. The blue producing compounds, right, produce a wound that deter the animal evolutionary from eating it. So it would be it would be a mycophagy response.
Yeah, so that's all speculative. So nobody's actually established that that's what's happening.
Right. But that's incredible. That you want to know.
Kind of thing I'm interested in pursuing. Yep.
So the other thing, the last question, Bryn, that we're going to answer was somebody who asked, who mentioned that they had found psilocybe on wombat and kangaroo scat, so dung. And so in terms of diversity of psilocybe, you mentioned there were 300 species more or less known. Do you think there are many more species? More species of psilocybe in the world than we know? Is there a large rate of discovery of new species?
Without a doubt, there are more species out there that we don't know about yet. I mean, fungi as a whole, as you know, we know about 5 to 10% of them. Mushrooms probably more than other groups. Lichens probably the best. Mushrooms probably second best. But I think it would be conservative to say that there's twice as many species of mushrooms out there than we know about currently. And that probably applies to most groups. Psilocybe, despite their interest, because they are intoxicating, they're really kind of inconspicuous, boring-looking things, and so they're probably often overlooked in surveys.
They're LBMs.
Yeah, they're LBMs. So I think there's probably even more of those out there than other groups.
So there, then Inocybe are also, and Gallerinas are also, we say FLBMs. You know why, right?
I couldn't guess.
What would you guess?
Funny little brown mushrooms.
No. Wrong answer. Try again. Try again. One more.
Oh boy.
Just say it.
Fucking little
you
brown
think
mushrooms.
It. Fucking little brown mushroom. Every time you see one, you're like, this is a fucking little brown, right? It's just full of fucking little brown mushrooms. So, and then gotta say it. I remember funky. We've got funky ones. Funky little brown mushroom, fantastic. Foundation, Fundación Funky little brown mushroom. Oh, we can all the FFs.
A competition for the most creative answer to that question.
Okay. Oh, I have to. Okay, I won't say. Oh, I have a good one, but I'll write that one later. To anybody out there. So one. So that was the last sort of. I wanted to ask you that question and say bye to everybody. Just before we go, I want to thank you, Bryn. I would continue talking for hours, but hold on 1 second. Sorry, we have Mushroom Gorilla. That's Antonio from Spain. Antonio, your account, you do all sorts of amazing days. He has Mycology Monday. He has Tattoo Tuesday. It's all fungal. He does like 3 posts a day, every day of the week with these themes. So Mycology Monday, Tattoo Tuesday, Wee Wednesday. The Thursday isn't done. And then Freak Friday, Artsy Saturday, and I don't know about Sunday. I've forgotten. So maybe he can help us do. So if you encounter a little brown mushroom in the woods, and you have to call it F something LBM, what would you call it? And we'll see what comes in from everywhere. Anyway, so everybody's thanking you. I just want to say hi to everyone, to the world. Mycology, 35 people are connected now, but we have hundreds watching, up to thousands. Slime Mold Sunday is the other one. Bryn, you closed the Myco Viral Night. Thank you. Can't thank you enough.
Thank you. This is super fun. This is what I'd like to doing all day. So I don't know. How do I close these things?
You've got to be funky and speak your heart. About
a mushroom a day keeps the doctor away. Keep the coronavirus away. How about that?
No, you close. Thank everybody, and we'll close, and I'll turn it off. So you've got the closing words. Go for it.
Thanks, everyone. Be strong. Eat lots of fungi. Drink wine.
Cordyceps. Take cordyceps drug. Thank you.
Cheers.
Ciao.
Ciao,
Ciao.
everybody.
Ciao.